Okay, so we are almost at the end of the quarter. So I just put up what you need to pay particular attention to. The last quiz is going to be this Wednesday, so that should be our 7th quiz. And you get to drop your 2 lowest quiz scores. So, 2. Yeah, 2 lowest quiz scores. I'll have your homework handed back. I'll hand back some homework and a quiz on Wednesday. But your last homework, which is on osmoregulation and hyponatremia, which means that you don't have not enough sodium in your body, that is due on Wednesday. So remember that your final lab report is due the 12th of March, and you need to submit that online. And so if we look at where that is in Canvas, so if you go to the home page, right underneath the home page is the formal lab report. So this is where you should be able to submit. So this has some instructions, right? And then if you're reading it as a student, you could, this just shows it as neat as I can edit it. You should be able to submit it from here, okay? And then we also remember that there is a grading rubric. So that would be helpful to look at so that you make sure that you get the full points. So this is the grading rubric. Your extra credit assignment is also submitted online. So this has some information about it. It needs to be something that we have talked about that relates to something this quarter. So if you start talking about like plants, and well, if it relates to animals in some way, you know, if you talk about herbivory and chemicals that the plants produce that interact with the nervous system of an animal, that might work. But we want to keep it to kind of some general topic that we talked about this quarter. But it should not just be a summary of what I presented. You need to go beyond what was presented in class. And so you also need to have references. So if you read an article and you summarize it, you need to make sure that you reference that in your extra credit assignment. So I'd recommend that everybody do extra credit, but I wouldn't recommend that you spend hours and hours on this, like maybe an hour tops, right? Because you want to spend most of your time studying for the final exam. So you don't want to put everything into this assignment when it's not worth as much points as the final exam. Okay, so question.
This might be a silly question. There's no way that taking 2 quizzes off is going to lower the person's score, is it?
No, because your quizzes are only worth 5 times 15. So it's only worth 75 or something like that. So in general, I mean, I guess it could possibly lower your score, but that's my policy, is to, so for those of people who missed a quiz, that's the general policy.
Is there any way that it will raise?
Yes, it will definitely raise people's score. So like, say for example, you got a 7 on one of your quizzes, that would be a good one to drop, right? So that's going to actually help your score overall. I think I've done my math wrong or something,
because it says that it lowers it.
Okay.
Do you like cut it out, or you make it like a 15 out of it? Grade book?
What I do is I cut it out. Yep. So what you'll get is I will take all of, on your grade book, I'll make all of the quizzes not count, and then I'll add up the 5 best quizzes, and then you'll get that score for your 5 best quizzes added into your overall grade. Okay.
Will there be a study exam for the last?
Yes. So I'll have I'll have a study guide as well. Probably on, probably next Monday. So the lab practical is not this week, but it is in 2 weeks. And so you want to make sure that you have the lab notebook put all together so that when you come in and you take the lab practical, you don't have to spend an hour, because that kind of gets annoying, putting everything together, like, oh, I have a piece of paper here, where does it go, right? You're supposed to kind of put it together as you do the labs. And so I'd like you to have kind of your lab notebook put together before coming to lab to take the lab practical. That will mean that you'll actually have some time to study, right? As you're putting your lab practical or your lab notebook together, you'll be able to kind of see what it is going to be on the lab practical. Okay, that lab practical is multiple choice. Okay. And then here I mentioned the extra credit as well. Okay. Are there any other questions about?
So if you missed a lab, do you put your extra credit inside your lab book?
No, you're going to submit your extra credits online, and then that will just go into your overall score. So your lab notebook might have a lower score than you want, but you'll have your extra credit, which will offset it when you get your totals. So I go totally by percentage of points, right? So everything is weighed exactly the same. I don't understand people who do. I was like, oh, your homework is only weighs 10%, but it has different, points. You can't really figure that out. So anyway, okay. Any other questions? Okay, so I have a handout to kind of review. So what is this structure called? What is that structure called? What is this? It's a nephron. And where is it located? In the kidney. And we have thousands, maybe tens of thousands. I meant to look that up, how many nephrons are in one kidney. We have a lot. Okay. So what does the nephron do? Yeah, that's what that does. Filters blood, right? What does it take out of the blood? Urea, right? Urea is our nitrogenous waste. What does it also do in terms of osmoregulation? What does osmoregulation mean? Yeah, so it regulates water salt solute content. So salt is a solute that is in our blood. So osmoregulation means that it takes energy for us to regulate our water solute concentrations. And I mentioned that some organisms like jellyfish are osmoconformers. So they're just whatever solute concentration is in the ocean, they just conform to that. But we are osmoregulators. Yes. Oh, you didn't. Did anybody else need one? I skip over anybody else? Okay. So does this look like a cortical nephron, or does this look like a juxtamedullary nephron?
Juxta would be running along the same as the.
How do I tell? Because it has a very long, what is this called? Loop of Henle, which goes down into the medulla of the kidney. So remember, if the loop of Henle does not extend into the medulla, it is called a cortical nephron. So these make up, the juxtamedullary nephrons just make up about 15% of the nephrons in our kidneys. So we write that as juxtamedullary nephron.
What was the name of the?
Versus a cortical nephron. Nephron. That's not a peace sign. That's an O. Okay. Nephron. Okay. So blood comes in, and where does filtration take place? So what is number 5 in your diagram? Blood is coming in, and what does number 5 look like? A capillary. And what is the capillary bed called in the nephron? The glomerulus. So number 5 is the glomerulus. Okay. Number 6 catches the filtrate. And so what do you think it would be? It's catching the filtrate that leaves that capillary bed. Then it is called the name of a person. Bowman's capsule. Sometimes that's also called a renal capsule. You do not need to know 7 and 8. So those are just small arteries that are carrying blood into the capillary bed. Okay, hence they're red in color. Okay, so what do you think 4 is? It is part of the nephron. It is the tubule that is right after the glomerulus. Proximal convoluted tubule. So you could abbreviate it as PCT. So that's my proximal convoluted tubule. Now you'll notice that there are blood vessels around the tubules. And so number 1, we didn't talk about these. These are called what are called the peritubular capillaries. So the peritubular capillaries in the PCT would be picking up things that are being reabsorbed. So like glucose. Glucose gets put into the filtrate, but we do not want glucose to end up in our urine. So what happens is glucose is pumped out and put back into the circulatory system. So the peritubular capillaries pick up substances that are reabsorbed. Oops, pick up substances that are reabsorbed.
Exactly what do you mean by that? So like pick up and then reabsorb or something, saying the same thing, or
Well, so there's actually channels. So this is lined with epithelial tissue, right? And so there's little channels, and they're little protein channels, and you might have learned about that in 211, right? So protein or channels that are proteins. And so they actually, if the glucose is really high in the urine, they actually have to use energy to pump it out. So they pump it out, which would be reabsorption. And so it's removed from, so reabsorption would be removing it from the filtrate. And then the capillaries pick that back up. So it's got to go out of the peritubular, the proximal convoluted tubule goes out of that, and then it's got to go into the capillaries. Does that make sense? Yeah, that makes sense. Yeah. So D, or number 2 is the distal convoluted tubule, because that is the furthest away. So notice that if we're looking at these little arrows, this shows the direction of the filtrate. So this arrow, for example, is showing that the filtrate is moving down into the loop of Henle. And then when it gets up here to number 2, this is my distal convoluted tubule. So a number 11 here would be what? What, we already talked about that. What is that long structure? 9 is actually the descending limb. So this is descending limb. 10 is the ascending limb. And these are all of number 11 would be loop of Henle. So 12 is another capillary bed, and it has a unique name. It is called the vasa recta. And it is specifically involved in concentrating the urine. So remember that when we talked about the loop of Henle, water moves out of the descending limb, and then salt moves out of the ascending limb. So the vasa recta kind of helps to maintain a solute concentration that is high in the medulla. Okay, so it's kind of picking up water and salt.
By osmosis,
water, osmosis. The salt is pumped. Yeah. So anytime it is pumped against its concentration gradient, it takes energy. And so this is actually one of the reasons why producing urine is energetically expensive. Specifically, if you're trying to produce concentrated urine. So if you're trying to like conserve water, it's going to take energy to do that. Okay. So number 3, this is the collecting duct. And you'll notice that more than one nephron attaches to a collecting duct. So all of these little branches right here, these would also be nephrons attached. Okay. So what we're going to talk about is the collecting duct and its ability to either be permeable to water or not. So we're going to say that the permeability of the collecting duct to water so that means the water can move across, is dependent on the presence of a hormone called the antidiuretic hormone. You can abbreviate this as ADH. So antidiuretic hormone, ADH. So what does diuresis mean? Like if you're diuretic, what would be a problem?
Leach out water.
Yes. So diuresis is getting rid of water in the urine. So you can take drugs and other substances that are diuretics. Okay. So one example of a diuretic is like vitamin C. So that's why they say taking massive doses of vitamin C actually could be detrimental because you have to urinate that extra vitamin C out. So it actually could cause dehydration. So taking massive doses of vitamin C might be detrimental. The other thing would be like caffeine and alcohol. So when you drink alcohol, like when you drink beer, you have to urinate a lot. And it's not just because you're getting rid of the alcohol metabolites in your urine. Alcohol is actually a diuretic. And so what happens is that you get dehydrated after you drink alcohol because of its activity as a diuretic. So a lot of the side effects of being hungover are actually dehydration. And so you can drink more alcohol and not be as hungover if you drink water while you're drinking the alcohol, right? So the kind of the rule is for every beer that you drink a glass of water. And then after a night of drinking, you force yourself to drink a bunch of water and you'll wake up feeling a lot better. I told my brother-in-law that when he was 50 years old, and he's like, oh, God, how come you didn't tell me that sooner? Anyway, so that's what you have to do. So it's not just the alcohol that makes you sick, but the dehydration that makes you sick. Okay? Okay, so antidiuretic, what do you think that does? What does the antidiuretic hormone do? Helps you to retain water. And it does this because we have protein channels called aquaporins. And they just recently discovered this, that these aquaporins are protein channels that, aquaporins are inserted, sorry. inserted into the membrane of cells lining the collecting duct. Okay, so what this means, it means that your collecting duct is going to become permeable to water, and water is going to move out of the collecting duct and be picked back up by the capillaries in that kind of surrounds the, maybe even the vas recta. Let's say it's picked up by the vas recta. So water moves out of filtrate and is picked up by capillaries. So ADH is actually released by your pituitary gland. So your pituitary gland is like in the center of your brain. It's kind of hanging down from your hypothalamus. And so there has to be a signal. And so your body has what are called osmoreceptors associated with the blood. And these osmoreceptors can detect solute concentration. Right? So it can detect the solute concentration. So if solute concentration is high in the blood, ADH will be released. So if we have an increase, it doesn't want me to let me do my arrow here. We increase solute concentration, that's a squiggle. Then we increase the secretion of ADH. ADH then has to travel via the circulatory system all the way out to the kidneys, all the way into the kidneys, where they can directly affect the cells that are lining the collecting duct and cause these channels, these aquaporins, to be inserted into the membrane, which allows water to move out and the urine to be concentrated. Okay? So likewise, if your solute concentration becomes too low, as in if you drink excessive amounts of water. So that was your homework assignment. So if you drink excessive amounts of water, then you're not going to release ADH. The aquaporins will not be inserted into the collecting duct, right? And then you're going to get rid of that excess water in your urine. But there's only so much water that your kidneys can process and can actually get rid of, so that you can actually literally drink yourself to death if you drink too much water. Yes?
Say, like, how do you know?
Usually you have to go, it's against your will, right? So if you like, the homework was like, I dare you to drink this amount of water. And you actually like have to like go past feeling sick, you know, you're drinking too much water. Yeah. The other thing is that you can sweat. And so if you're a person that's working outside and you're sweating a lot and you're drinking water, you actually have to take salt pills or drink something with salt in it. You can drink salt water. But like if you're like working outside all day long and just sweating, then they recommend that you take salt pills.
Or would you drink like Gatorade?
Gatorade, yeah. Yep. Pedialyte. Yep. Yeah, that is what's good for hangovers too. Pedialyte. Yep. Okay. Any questions about that? So this is a way that we can regulate our osmotic concentration in our blood through the use of a hormone. Okay. So let's talk about the urinary system. So this obviously includes the kidney or the kidneys. The kidneys then collect to or connect to a ureter. So a ureter transports the urine to the bladder. So the bladder just simply functions in the storage of urine until you have the opportunity to get rid of that urine. The bladder is actually composed of smooth muscle. So the bladder stores urine. And so that kind of means that it will contract involuntarily. So one of the really interesting things about having to urinate, that sensation, is that when you feel like you have to pee, right, that is actually the smooth muscle of your bladder contracting involuntarily. So you're like, oh, I have to pee. And then you say, well, just wait a little while, right? This happens all the time on car trips. You're like, oh, I don't have to pee now, right? Because if you don't urinate after a while, the smooth muscle will stop contracting and all of a sudden you don't have to pee, right? And then 5 minutes later, you're like, I have to pee now, I have to stop, or you have to stop so I can pee now, right? And that's because it will just keep contracting and relaxing until you give it the signal to open up the valve, essentially, the smooth muscle valve that controls whether or not urine will come out or not, right? So sometimes if you don't urinate, what do they do to you in the hospital? Right, so they can put in a catheter. So a catheter is used to drain the bladder. Right? So that's a tube that goes up the other tubule, which is called the urethra. So the urethra drains the bladder to the outside of the body. So the catheter actually goes through the urethra. So in males, that is a long tube that comes out through the penis, right? So this, in males, transports both urine and semen. And we'll talk about what semen is. It's sperm plus all the secretions of the seminal vesicle and the prostate gland. Okay. So it is a long tubule in males, but in females, it only transports urine. So females actually have 3 openings that are relatively close together. That would be the opening to the urethra. That would be the opening to the reproductive tract, which would be the vagina. And then that's the opening to the digestive system, which would be the anus. Males only have 2 openings. They have the urethra opening out and the anus, right? So sometimes the urethra can get infected, and you have what is called urinary tract infections. Or UTIs. And this can be due to bacteria getting into the urethra, and then it can actually travel to the bladder. And if untreated, it can actually get up into the ureters and travel to the kidneys. And that makes you very, very sick if you get a kidney infection. Right? So you get like a high fever, you have to go to the hospital, they have to put you on some super duper antibiotics. So generally, urine is sterile. Right? So it should not have any bacteria in it. So when you go to the doctor and they ask for a sample of your urine, what they're doing is they're trying to determine if there's any bacteria, or sometimes there's white blood cells as well, but the urine will appear cloudy, and that suggests that it has bacteria in it. Right? So these generally are due to bacteria, so then they're used, you get a antibiotic to treat them. So if we look at these images, right? So this is from your OpenStax online book. So here's the kidney. Notice that there are major arteries and veins leaving the kidney. The renal artery carries blood in, the renal vein carries blood out. This is the abdominal aorta, and this is actually the vena cava. So then it branches. We won't talk about the. Let's talk about branching into the iliacs now. Okay, so let's just talk about the kidneys. So this is the ureter. So notice the ureter carries the urine to the bladder, and then from the bladder we come out via the urethra. So that is what is referred to as the urinary system, because it contains multiple organs that all aid in osmoregulation as well as getting rid of nitrogenous wastes. Are there any questions about the urinary system?
So the urethra, it's just like basically a tube for your urine to transport to?
Yes. Yep. It's just a tube, and there's a little sphincter that you can control. You know, we know when we want to void, right? Sometimes that sphincter becomes weak, and so sometimes people have problems when they get older, specifically as they leak urine, which can be annoying. Right? So keeping that pelvic floor strong can sometimes help prevent unwanted leakage of the urine out through the urethra. Okay, so that's it for osmoregulation. So we are actually moving to the nervous system. So we've already talked a little bit about nervous systems. What is the first organism that we talked about that has a brain?
Gastropods.
What'd you say? I didn't hear you.
I
Sorry.
just said gastropods.
Gastropods do have a brain, but they're not the first. Think back, like when the first, it's within the first three phyla. What's the first phyla that has a brain within the first three groups? So group number one we talked about, what were the first animals we talked about? Sponges. What were the second animals we talked about? Cnidarians. What were the third animals we talked about? Platyhelminthes, right? So this is like your planaria, right? So platyhelminthes. Okay, so the planaria. They have cephalization. So they have a brain. They also have specialized sensory structures called eye spots. And then they also have auricles, which look like little ears, but they use actually to navigate and to orient towards chemicals in their environment to essentially find food. Okay. So before that, when we were looking at the cnidarians, what type of symmetry do the cnidarians have? Radial symmetry, right? So they have no cephalization. So we see no central aggregation of nerve cells. So they have no brain. Rather, they have what is called a nerve net. So the neurons are equally distributed all around in their body. And that allows them to move in any direction, right? So they don't have to move through into the new environment head first. So if we look at some examples of nervous systems, these would be the cnidarians. Echinoderms. The adult echinoderms also have no brain. So remember the sea stars, even though they can have pretty complex hunting behaviors where they use their 2 feet to pry open clams and other mollusks, they have no brain. When we looked at the planaria, they have these nerve cords that extend. The arthropods, including the insects, have a ventral nerve cord, and it is solid. The cephalopods are known to have a very large brain, and they have very large neurons. Okay? And then, obviously, we are vertebrates. And what type of nervous system do we have? What is our characteristic nervous system of a chordate? spinal cord, but what is it called when it's... So the notochord, remember, is not nervous system. It is actually like skeletal. So it's the hollow dorsal nerve cord. So vertebrates, all vertebrates, right, have a hollow dorsal nerve cord. So that was a very different type of nervous system than is seen in the rest of the animal kingdom. So if it is hollow, what is it filled with? Fluid, and what's that called? Cerebro, well, there's actually spaces in the brain. So we'll say that it is filled with cerebrospinal fluid. And this has a abbreviation. You could call it CSF, cerebrospinal fluid. So even in our brain, we have spaces called ventricles that are filled with fluid and help to bathe the tissue. Right down the middle of our spinal cord is a hollow space that is filled with cerebrospinal fluid. And that fluid actually circulates. It's produced up in the brain. It circulates down your spinal cord, and then it has to come back up and be reabsorbed. So your cerebrospinal fluid is always being produced and reabsorbed, and it's derived from blood. So there's specialized capillaries that aid in the production of cerebrospinal fluid. So if your cerebrospinal fluid starts to leak out, and sometimes that happens if they try to take a lumbar tap, if they try to take some cerebrospinal fluid, sometimes the fluid will leak out and you'll have low cerebrospinal fluid. And what that generally causes is intense headaches. So sometimes people have to be hospitalized and they have to lay on their back because if they sit up, they don't have enough cerebrospinal fluid in their ventricles of their brain, and it'll be extremely painful. And so then they have to give you fluid to make up for that which has been lost from the spinal fluid. I know that sometimes that happens when women get an epidural, which means that they get an anesthetic inserted into their spinal cord to block pain. And so sometimes women wake up and they're like in extreme amount of pain because some of their cerebrospinal fluid leaked out.
Does the body regenerate that?
It does regenerate it. So you can wait, but sometimes they give them what is called a blood patch. And the blood patch just kind of patches it with some derived, they actually kind of synthetically make it and put it back in. Yeah.
Is that fluid sterile?
Yes. So if you get so sometimes they'll take a sample to see if there is an infection in there. So meningitis can sometimes lead to infection that gets into that, and that can be deadly, right? Yeah. So you want it to be sterile.
Is meningitis bacterial?
Meningitis can be both viral and bacterial, and it's actually inflammation of the meninges that cover both your spinal cord, it's connective tissue coverings of both your spinal cord and your brain. Yep. Okay, so we're actually going to talk about today, not the nervous system itself, but we're going to be talking about what makes up the nervous tissue, and we're going to be talking about the physiology of this. So this is on the back of your handout. Okay. So this is an example of a neuron. So not all neurons look like this, and I'll show you a picture of more complicated neurons, but this just looks like a typical neuron. Okay. So we see that there is in the center of the cell a round object that is dark, stains very darkly, and what would that be? That's my nucleus. Okay, so this part is the cell body. And this is where the organelles are found. And one of the things that the cell body has to produce would be neurotransmitters. And so the neurotransmitters are produced here. Okay. Okay, so notice that we have this cytoplasmic extensions that are highly branched. So does anybody know what those are called? Dendrites, excellent. So you're like, where does that word come from? The study of branching and also like the way that trees branch is called dendrology. So dendrites are like the branches of a tree, okay? The dendrites receive information from other cells. And the way that they do this is that they have receptors that bind neurotransmitters. Okay. Let's make sure I'm recording. Okay. Okay, so you'll now notice that there is one long cytoplasmic extension. And what is that called? Does anybody know? So this long cytoplasmic extension, which they're actually showing here, is the axon. So I'm going to write what the axon does over here to the side. Okay. So the axon transmits information to the ends of the axon, which are called the axon terminals. One of the interesting things about nerves, for example, nerves are actually bundles of axons, right? So when you talk about your sciatic nerve, what's really interesting is that the cell bodies actually reside in the spinal cord. And the nerve that extends out from your spinal cord and all the way down your leg, the sciatic nerve, is just made up of bundles of axons. And so we would say that neurons are actually the longest cells in the body. So they are longer than muscle cells because they have these cytoplasmic extensions that extend all the way out. Okay. Okay. So these right here are the axon terminals. Now the yellow structures on my diagram act as insulation. And there are two different types of cells that insulate the axon. So these could be Schwann cells. And so this is if they're in the peripheral nervous system. So like if we're talking about the nerves that extend out to the body, they're Schwann cells. If they're in your brain or your spinal cord, they're called oligodendrocytes. So this would be in the central nervous system. What does oligo mean? So earthworms are oligochaetes, few dendrites, okay? Actually, they act as insulation. So it actually speeds up the transmission of the information. We're going to talk about what that information is in a minute, okay? So the spaces between the Schwann cells have a name, and they are called the nodes of Ranvier, after a person. That's a V. Ranvier. Let me spell it again. Okay, Ranvier. So we'll talk about the significance of that. So what is the space at that would be between the axon terminals? And if I was going to like draw another neuron here, so like say that's another neuron, what is this space called? What is the space from one neuron to the next called? Anybody know? Synapse, yes. So this is the synapse. Right? And so when they say when you learn, you create new synapses, right? So you have synaptogenesis, which is the creation of new connections in the brain from one neuron to the next. Okay? So the information has to get across that synapse in some way. And we're going to talk about how it does that today. Okay, so I just wanted to point out that that was just a kind of a classic drawing of a neuron, but neurons are very, very diverse. And so we see different structures of neurons depending upon whether they are sensory. Like, for example, if they're carrying information about touch into the nervous system, or whether they're transmitting information from the sensory part to the motor part, which would be the interneurons. And then the motor neurons actually kind of look like that. They actually have that central body in one long axon. So this would be like the motor neurons extending out to your skeletal muscle, causing them to contract. Okay. So in your book, this is another picture of in your book. And one of the important things to realize about nervous tissue is it is just not made up of neurons. So nervous tissue is composed of neurons plus glial cells. These glial cells are what we call helper cells. So when I was studying, we didn't even realize the significance of the glial cells. But what they have discovered is that for every one neuron in your brain, there is about 10 glial cells. So the helper cells actually outnumber the neurons 10 to 1. So they must be doing something significant. So they call them helper because they help to maintain the environment in which these specialized neurons reside. And so the oligodendrocytes, this must be the central nervous system, right? Because that's the oligodendrocytes. We also have astrocytes, which you don't need to know the different glial cells, but the astrocytes actually connect the circulatory system to the neuron. And so they actually create kind of the blood-brain barrier between our nervous tissue and our blood. And then these microglial cells have become really important recently because they actually think that microglial cells can go in and can remove debris. So what's an example of a disease where there are plaques of proteins that are not supposed to be there, that are interfering with the way that one neuron communicates with another? What's the disease where they have this extra protein? Specific type of dementia? Alzheimer's, right? So they're thinking that these glial cells could actually go in and clean up the environment, remove the plaques so that the neurons could be more healthy. They also think that these glial cells are important in sleep. One of the things that happens when people get Alzheimer's is that they stop sleeping properly. And you have a lack of sleep, what they think of what happens when you sleep is that it's almost like garbage day, right? So they think that these microglial cells, kind of the blood vessels kind of open up in the brain and there's more space, and the microglial cells go in there and they clean out the debris between the neurons so that the neurons will work properly. And so microglial cells might prove to be really significant in the study of dementia and Alzheimer's. Okay. So let's talk about the physiology. So what we're talking about here is the mechanism, not the anatomy. So what is the mechanism of a nervous impulse? So if we look at neurophysiology, we can talk about an impulse generation. And this is the same thing as what is called an action potential. So this is an impulse that gets generated. And if we look at potential here and we start talking about what a potential is a difference in the charge between the inside of the cell and the outside of the cell. So we can talk about the membrane potential. And one of the really weird things about our body is that it is electrical in nature. It has this electrical nervous system. And so when we talk about potentials, we're talking about the same thing that you study in physics, right? So the membrane potential is a difference in charge between the inside and the outside of the cell. Now, in chemistry, what are charged particles called? Ions, right? And so this is due to differences in ion concentrations. And the important ion concentrations are sodium, K is potassium, and chloride. Sodium, potassium, and chloride are our ions. Okay, so I'm going to draw the inside of my axon. So this is my axon. This is outside so this would be like the interstitial fluid, and this is inside. So what I'm going to do is I'm going to draw it when it is not generating an impulse. So we're going to talk about the resting potential. So this is my resting potential. And it has a difference in charge of negative 70 millivolts. So when my axon is doing nothing, the difference between the inside and the outside is negative 70 millivolts. And so what that means is that the inside has a negative charge, right? So I'm going to draw my negative, let me use different colors, I'll make it prettier. Negative, negative, all along here, and on this side too. And then positive on the outside. So they actually have these little tiny micro voltmeters that they can insert into the axon and actually measure the difference in charge. Same thing you would do if you were trying to see if electricity was being conducted in a wire. Okay. Okay. So on the outside, we have lots of sodium. So I'm going to put sodium in big, and I'm going to put sodium in real little on the inside. It's exactly the opposite. We have potassium real big on the inside, potassium real small on the outside. And then chloride is more concentrated on the outside and more small on the inside. And then stuck in the axon are large proteins, not large proteins, but actually they're small proteins, but they're large anions. So this is a negatively charged protein. Okay, so anion, that's why it's an A. So anion versus a cation. Anion is negative, cations, ions are positive. Okay. Okay, so these charged particles actually they can move. So the membrane is permeable. So how would the charged particles, you think, how would they move? With sodium, how would sodium move? It would move in because things move against their concentration gradient. So you'd predict that sodium would just diffuse in until an equilibrium was established. And you think that potassium would just diffuse out until an equilibrium was established. And the same thing with the chloride. The anions cannot move. They get stuck inside. So that negative charge will be important when we talk about why it goes from negative to positive. Okay. So the resting potential is maintained. So the ion concentrations are maintained via what is called the sodium potassium pump. Right? So sodium, when it does diffuse in, is pumped right back out. Potassium, when it diffuses out, is pumped right back in. So this takes energy. So it requires a channel. So that would be like an opening, and it takes energy. Energy in the form of ATP. So the significant thing about this is that even at rest, when our nervous system is doing nothing, it takes energy to maintain it, right? This is why animals will reduce their nervous system, the number of neurons they have in their brain, if they do not need them, right? So if you don't use it, you lose it, right? You reduce the number of neurons in your brain if you don't use it. And so this is why they just recently discovered that animals like chickadees are these little birds that overwinter here and overwinter in the north, and they cache food all around. And in the winter months, they actually have to have a bigger brain to remember where they have stored all of their food. So their hippocampus is actually quite large in the winter. And then when they go into summer, their hippocampus shrinks and contains less neurons. So that in the summer, when they don't have to remember where their food is hidden, they don't need as big a brain. And so they're actually discovering that animals have the ability to expand their brain when they need to learn, and then their brain shrinks when you don't need to learn. Unfortunately, one of the things that causes our brain, our hippocampus, to decrease in size is stress. So if you're really stressed out, there's a reason why you can't remember stuff. And that's because your hippocampus will actually shrink and you'll have less neurons available to remember stuff. Yeah.
You know, there's research done about like when you're training animals, the brain is bigger and then they were to stomp the trees.
I don't know of that specific. I do know that they recently discovered, they couldn't figure out why corvids, which are like blackbirds, magpies, crows, ravens, how they could be so intelligent and have such tiny brains. And so what they've discovered about birds is that their neuron density in their brain is much higher than ours. So their brain is smaller, but they actually have more density per unit of mass than our brain. And so they have a lot of neurons, it's just packed in a more tight space. So I'm not sure. That would be interesting to see. They have definitely done stuff on the hippocampus, but I don't know about training animals. Yeah. Okay, so in your book, there is a diagram that shows the sodium-potassium pump. Okay, the first one, you could take a microelectrode, stick it inside of a neuron, and you can see the difference in charge between the inside of the neuron and the outside of the neuron. So you'd probably see this going on if you went in, if you're at a university and you went into a neurophysiology lab. They would probably have some equipment set up to measure voltage within neurons. So the resting potential is a negative 70 millivolts. Here, we're just paying attention to the potassium and the sodium. So we'll talk more about chloride in a little bit. But notice that energy is required to take that potassium and pump it in, and then the sodium is pumped out. Okay? So that requires energy to maintain the difference in concentrations. Okay, so we're going to take maybe 5-minute break, and then we're going to come back and we're going to talk about how your body generates an action potential. And we'll talk about what this graph represents as well.
Well, I drive my dad's car to school, but as around town, I drive by also. But I can, it's got a gas.
Okay. So resting potential was negative 70 millivolts. Your action potential is a positive 40 millivolts. So if I were to draw what this looks like in my axon, right, it starts out with it being negative, and then here's the positive, and then we get a reversal. So here we have positive on the inside and negative on the outside. And so I would say that this is where the action potential is being generated right here. So before and after are going to be normal. So it's a reversal in the charge. So for some reason, the inside becomes positive, so the charge has reversed. And that is actually what's drawn on your handout. So if we look at the handout, it looks something like this. This handout, just put, sometimes they have positive 30, so just put positive 40 on this. One in your book has positive 40. Okay, so here, this negative 70, we're going to label this as the resting potential. So that is where no information, nothing is being transmitted from one neuron to the next. Okay, it's at a negative 70. And then this little red arrow here is the stimulus. So that could be the binding of a neurotransmitter that starts to open up ion channels. Okay. Now notice it goes from negative 70 up to 0. So this process of going from negative 70 up to 0, this is called depolarization. So depolarization. So think about polarized means negative 70 and a positive. That would be a big difference, a polarity. So depolarization is going towards 0, and then notice that it overshoots 0 a little bit and comes up to a positive 40. So the opposite, where it comes back down, this would be repolarization. And if it goes below the resting potential, it is said to be called hyperpolarization. Now the dotted line is important because this is my threshold. So if the stimulus is too weak, it could cause a partial depolarization, but if the depolarization doesn't get past the negative 55 millivolts, then an action potential will not be produced. So the important thing to realize is that all action potentials are identical. They're all the same strength. Okay, so all action potentials are identical. So what that means is that stronger stimuli can be sensed because not due to a stronger action potential, but due to the fact that more action potentials are produced in a given period of time. So a stronger stimuli will generate more action potentials. So the difference in, like, if you were to listen to a loud sound versus a whisper, the reason why it sounds loud to you is that there's more action potentials being initiated in your cochlea and then sent to via your cochlea nerve to your cerebral cortex, where you consciously sense the sound. Okay. So all action potentials are identical. They're also all or none once the threshold has been met. Oops, has been met. Okay. That kind of goes along with them being identical. So you don't have like weak action potentials. They're all or none, as long as that threshold has been met. Okay, so what causes this change in polarity? It has to do with gated ion channels. So this means gated means that they're not just open. That means that some of the time they are closed and some of the time they are open. So they can be opened and closed, and that's what it means to be gated. So we have voltage gated channels. And these would be like in the axon. So once an action potential is generated, it travels like a wave down the axon, and it opens voltage-gated channels to allow ions to flow. But we also have chemical-gated channels. So, for example, some of our neurotransmitters will actually bind to receptors that are attached to a channel. And if the chemical binds, it's going to open it up. And so this would be like in the dendrites. Okay. So during depolarization, sodium-gated channels open. We've been pumping sodium to the outside. So what happens is sodium floods in. So sodium floods in. Right? And so that flooding in of sodium is going to bring the positive charge with it. So it's going to make the inside positive, a positive 40 compared to the outside. Okay. During repolarization, these sodium channels close, but potassium-gated channels open. So potassium, we've been pumping to the inside. So then potassium floods out. And so by taking the positive charge with it, the inside becomes repolarized and becomes negative. And it generally overshoots repolarization, so it becomes hyperpolarized before it hits back to resting. And so that means that there's going to be a delay before another action potential can be generated. Okay. So the weird thing about this is notice that we haven't talked about the movement of a negative charge particle at all. They're just positive charge particles, but their opposite movements cause a change in the polarity of the membrane. Does that make sense to everybody? Sorta? Okay. So in your book, I would advise you to look at this diagram. Okay. So notice that this is a gated channel. Notice that this is not a gated channel. So that channel that is not gated, sodium is just going to move across, right? Because the diffusion always kind of takes place. Okay. So when it goes to here, notice that sodium starts to flood in. So that sodium channel is going to open. Okay. And that sodium channel here is going to close. So my sodium channel closes upon repolarization, and the potassium channel opens, and so the potassium will flood back out during repolarization. Why is it negative charged in the first place? What's giving it the negative charge in the first place? The big anions, right? The big A, remember, which does not move. So the relative movement of the positive charge particles is what causes the difference in charge. Does that make sense at all to anybody? Something you kind of have to think about, I think.
So they're all positive, and the A is what?
This is negative, and it is stuck inside the neuron. There are no channels for it. So that's why when potassium floods out, it becomes negative. And that's the resting potential is negative 70 millivolts. Did you guys have a question?
Does chloride always stay on the inside? Or
No, we haven't talked about chloride yet.
Okay.
So we'll talk about chloride maybe next. Okay, yes. So we're going to talk about synaptic integration. Okay, so this is the idea that one neuron is not just connected to one single neuron, but you have many neurons that are synapsing with a single neuron. Like you could have one neuron that is synapsed with 1,000 other neurons. And so it's receiving all kinds of information, and that information needs to be integrated. Okay, so one neuron receives multiple signals. Okay. Some of these signals are excitatory. So they call this the excitatory post. I hate this. The way that oh well, I'll just write it. I hate this terminology, but synaptic, excitatory postsynaptic potential. So it's the EPSP. Okay, EPSP, excitatory. Okay, notice the green there. So excitatory means that it moves the membrane towards the threshold. So does anybody who has taken psychology, can you give me an example of a name of a neurotransmitter? Of a specific neurotransmitter in the brain, for example? What's the one that you, when you're enjoying yourself? The feel-good one. What's it called? Dopamine. Okay. So dopamine is an excitatory neurotransmitter. Sometimes we take drugs like cocaine that increase dopamine levels for short periods of time. Right? We listen to music. I just read this. When you listen to music, it causes dopamine to be released. And so it's got that buzz that you get from listening to your favorite music is due to dopamine. So music is like being on drugs, I guess. That's why the teenagers are always all night long, really, with their headphones in.
Yes.
You do? Oh, God, it would drive me crazy all night long. My kids listen to music all night long. Okay, so dopamine. So this is moving it towards the threshold, right? The opposite of this is the inhibitory postsynaptic potential. So it's a good thing that we don't just have excitatory neurotransmitters. We also have inhibitory neurotransmitters. So these would be inhibitory neurotransmitters. So what is the inhibitory neurotransmitter in the brain that decreases anxiety if it's present in abundance? Yes. So serotonin. Okay. So serotonin balances out the dopamine. It makes you mellow, right? So if you take serotonin inhibitor drugs, reuptake like Prozac or Celexa, I had an interesting thing happen to me. So there was this guy colleague, a fellow teacher, he used to know how to push my buttons. He would say things and I would just like, oh. And so I had started taking Celexa, which is one of those mood stabilizers, right? And I was doing my copying and he came by and he said something. And I'm like, I don't feel anything. I'm not angry at all. And I just like, and he looked at me like, aren't I going to get a response out of you? And I'm like, no, not today, because I didn't have any effect on me. Okay, serotonin, mellow, right? No getting upset. People can say stuff all they want and just kind of stay mellow. Right? The problem with serotonin is that it can also affect other aspects of your biology that are not so good. Okay, so this is where the chloride comes in. Okay, so when we talk about inhibitory neurotransmitters, this opens up chloride ion channels. How is chloride going to flow? In. Right? And this moves the membrane away from the threshold. Okay. So notice here, boom, goes back to negative. So that's the significance of the chloride. This would be sodium flowing in, this would be chloride flowing in. Sodium flows in enough that it reaches that threshold, and this threshold is, says negative 10, should be negative 5. Then it activates the action potential, and then this is hyperpolarization, and then back to resting. Okay.
So all of those curves, I'm looking at the graph that we have before this.
Yes, there's none of that. Yes, and so this means that this neuron is receiving information from all kinds of other neurons, but it's not responding until the threshold is met. So that one could be receiving information. This just is what shows that integration. And so if you have too much dopamine, you're going to be overexcitable. If you have too much serotonin, you're going to be underexcitable. So there's a balance between your excitatory and inhibitory neurotransmitters that your body tries to maintain so that you're not overreacting to stimuli or underreacting to stimuli. Any questions about that? I know that's kind of a lot. Neurophysiology there.
What if you take it psychology?
Yeah, if you've taken psychology, you probably all heard this all before. So you're at a benefit. Okay, let's just talk about one more thing before we finish today, and that's the conduction of the action potential. So the conduction of the action potential depends upon whether you have an unmyelinated axon. Oops, spell it right. I need to figure out how to erase with my pen. Okay. So think myelin is the insulation. So myelin is insulation. And so if I was to like draw a cross section of my axon, so if this is my axon, the way that the myelin works is that you have this cell, this glial cell, that wraps itself around the axon, just like electrical tape. So you know that black electrical tape? You wrap around a wire that is exposed, right? So that it doesn't zap you. So that blue part, the purple part is the axon, the blue part is the Schwann cell or the oligodendrocyte, okay? So the unmyelinated axons do not have this. So this is an example of an unmyelinated axon. So we'll say that there are no Schwann cells. No oligodendrocytes either, but it is more significant with the Schwann cells because of a particular disease that attacks them. Okay. And the movement of the action potential is a slow wave of depolarization. So the action potential moves like a wave down the length, and then when it gets to the end, it can cause neurotransmitters to be released into the synapse with another neuron. So in your book, they have this picture that shows the action potential, right? So this in green, this is my action potential. It's being depolarized. Notice that it's got a positive charge inside, okay? So after it passes, the membrane goes back to the resting potential, okay? This is different than myelinated neurons. So myelinated axons have Schwann cells. And the action potential jumps from node to node. And this is faster. So when babies are first born, you'll notice that they're kind of jerky in their movements and they're not really coordinated and their movements are real slow. They tend to be real slow. And that is because when we're born, a lot of our axons are not yet myelinated. And then as we develop, the axons become myelinated. And then our movements can be real fast, right? And they can be real controlled and quick. So what is the disease that is a demyelinating disease? It's an example of an autoimmune disease where a person's immune system will attack and destroy the Schwann cells. Not Parkinson's. Similar maybe. So a person loses the ability to walk, they end up in a wheelchair, and they can slowly, it starts with an N. So multiple sclerosis. Okay. So this is where you have the immune system attacks Schwann cells. And so this is called a demyelinating disease. So the interesting thing is that by looking at this disease, we now know how significant the Schwann cells are, right? If you don't have the Schwann cells, your movements become real jerky, you lose the ability to walk, right? So that the Schwann cells are really important. Now, there's no cure for this one, but there's one that is very similar to multiple sclerosis that sometimes will cure itself. And so let me just get the name of it because I can never figure out how to spell it. That's, you know, using Google as your spell checker. Okay. So it's called Guillain-Barré. So if somebody wants to do this for their extra credit, kind of really interested in this. I don't know why I'm interested in it. I think it's fascinating. So what this is that you'll get a really bad virus, right? Maybe a flu virus. And you'll start to feel better. And then all of a sudden, one day you'll wake up and you're paralyzed and you can't move. And they take you to the hospital and you're in the hospital or a rehabilitation center for like 6 months. And you slowly regain the ability to move as your neurons become remyelinated. But it takes like a year or maybe even 2 years for this remyelination to occur. So somehow your immune system freaks out by having this virus inside of you and destroys your myelin almost overnight. And this can actually happen in dogs too. But generally they would just put the dog down because who's going to want to take care of a paralyzed dog for a year, you know, before they can learn how to walk again. Anyway, but they do this for people. Right? So that's another one. This one tends to cure itself, right? So you regain the ability as your myelin is replaced. Okay, so I will stop there today and we'll have a quiz on Wednesday over the urinary system, the kidney, nephrons, and then also what we talked about today. And that'll be our last quiz.